Welcome to G Fun Facts Online. Today, we're diving deep into a microscopic war happening inside the human body, a battle where the enemy uses a surprisingly sweet disguise. We're talking about cancer, but not just its uncontrolled growth. We're exploring its most cunning defense mechanism, a cloak made of sugar. Forget what you think you know about cancer and sugar being just about fuel. This is a story of sweet deception, where cancer cells weaponize sugar molecules to build fortresses, hide from our immune system, and actively sabotage our body's defenders. This is the cutting edge of cancer research, where scientists are learning to crack the glycocode and turn cancer's greatest strength into its fatal flaw. Every single cell in your body, healthy or not, is coated in a dense, complex layer of sugar chains called glycans. This sugar coat, known as the glycocalyx, isn't just decoration. It's the cell's ID card, its communication hub, and its interface with the outside world. It's how your immune system knows to leave your healthy cells alone. It reads this glycocode to distinguish friend from foe. But when a cell turns cancerous, this sugar coating undergoes a dramatic and sinister transformation. This process, called aberrant glycosylation, is a universal hallmark of cancer. The sugar chains on cancer cells become mangled. They're often shorter, overexpressed, or decorated with bizarre structures not found on healthy cells. These altered sugar structures are called Tumor-Associated Carbohydrate Antigens, or TACAs. And they are not passive bystanders. They are active weapons in cancer's fight for survival. So how does cancer use the sugary armor? The first line of defense is physical. In many cancers, the glycocalyx becomes incredibly thick and dense, forming a literal nanoscale fortress. This isn't just a side effect. It's an actively maintained shield that cloaks the cancer cell. Research shows a direct link. The thicker a cancer cell's glycocalyx, the harder it is for our immune cells, like natural killer cells and T cells, to kill it. Even a change in thickness of just 10 nanometers can be the difference between life and death for the cancer cell. This physical barrier literally keeps immune cells from making direct contact. It hides the very protein antigens that T cells are trained to recognize, preventing our body's most powerful adaptive immune response from ever getting started. A key ingredient in this fortress are mucins, which are huge, heavily sugared proteins. In many cancers, mucins like MUC1 and MUC16 are overproduced. They extend far from the cell surface, creating a dense, brush-like layer that can be up to 20 micrometers thick. This creates a slippery, impenetrable halo that not only pushes immune cells away, but also blocks chemotherapy drugs from reaching their target. Beyond just being a physical wall, this sugar coat is a master of disguise. It actively masks the cancer cell's identity. Healthy cells present antigens on their surface using molecules called the major histocompatibility complex, or MHC. This is how they signal to T cells if they're infected or cancerous. But the dense forest of glycans on a cancer cell can physically cover these MHC-presented antigens. The cancer cell is effectively hidden in plain sight, invisible to T-cell surveillance. But this sugar shield isn't just a passive defense. Cancer cells use their altered glycans to actively sabotage the immune response. They speak a sweet language of immunosuppression. They do this by interacting with specific sugar-binding proteins called lectins, which are found on the surface of our immune cells. These interactions trigger off switches within the immune cells, disarming them and creating a safe zone for the tumor to grow. One of the most significant changes is a massive increase in sialic acid, a type of sugar, on the ends of the glycan chains. This hypersialylation is a common feature across many cancers. Sialic acids on the cancer cell bind to a family of lectins on immune cells called Siglecs. This binding sends a powerful inhibitory signal. It's essentially a molecular "do not eat me" sign. This has devastating effects across the immune system. For our T cells and natural killer cells, this interaction shuts down their activation and their ability to kill. for our dendritic cells, which are responsible for training T cells to fight cancer. This interaction keeps them in an immature state, preventing them from raising the alarm. And for macrophages, versatile immune cells that can either fight or help tumors, this signal flips them to a tumor-promoting M2 phenotype. These M2 macrophages then help the tumor build new blood vessels and further suppress the immune response. Another key player in this sabotage are the galectins, another family of lectins that are secreted by tumor cells into the surrounding area. Galectin-1, for instance, can kill activated T cells outright. It also promotes the creation of those ineffective tolerogenic dendritic cells and helps the tumor build its own blood supply. Galectin-3 and Galectin-9 are also major culprits, helping to create an environment of immune tolerance where the cancer can thrive without fear of attack. Let's put a face to some of these villainous sugar molecules. The TACAs themselves have names. For instance, there are the TN and sialyl-TN antigens. These are basically stunted, incomplete sugar chains that are rare in normal tissue but pop up all over many cancers. They interact with our immune cells, like macrophages and dendritic cells, preventing them from maturing and doing their job. Then you have sialyl-Lewis A and sialyl-Lewis X. These structures are famous for their role in metastasis. They act like grappling hooks, helping cancer cells stick to the walls of blood vessels so they can escape and form new tumors in distant organs. Another important group are the gangliosides, like GD2 and GD3. These are shed by tumor cells and flood the tumor microenvironment. They are masters at suppressing T cell activation and dendritic cell function. So where do these faulty sugars come from? They are the direct result of a hijacked cellular factory. The enzymes that build sugar chains, the glycosyltransferases and glycosidases, go haywire in cancer cells. The delicate balance is broken. Some enzymes that add sugars, like those adding sialic acid, are cranked into overdrive. Others, which would normally build longer, healthier chains, are shut down, leading to truncated structures like the TN antigen. Understanding this dysregulated machinery is key, because these enzymes themselves are now becoming targets for new cancer drugs. Perhaps one of the most exciting discoveries is how this sugar coat interacts with the revolutionary field of cancer immunotherapy. You've likely heard of immune checkpoint inhibitors, drugs that target pathways like PD-1 and PD-L1. PD-L1 is a protein on cancer cells that binds to the PD-1 receptor on T cells, delivering a stop signal that shuts down the immune attack. We now know that this entire process is controlled by sugar. PD-L1 itself is a glycoprotein, and its sugar coating is essential for its function. The specific glycans attached to PD-L1 protect it from being degraded by the cell. This keeps high levels of PD-L1 on the cancer cell surface, maximizing its ability to suppress T cells. This means cancer uses sugar to stabilize its primary defensive weapon against our most advanced therapies. This discovery opens up incredible new possibilities. If we can develop drugs that stop PD-L1 from being properly glycosylated, we could destabilize the protein, making cancer cells visible and vulnerable to T cell attack once more. The good news in all of this is that this sweet deception is also a profound vulnerability. Because these tumor-associated carbohydrate antigens are so specific to cancer cells, they make for excellent targets. Scientists are now developing a host of strategies to exploit cancer's sweet tooth. First, there are antibody-based therapies. Monoclonal antibodies can be designed to specifically seek out and bind to TACAs on cancer cells. Once bound, the antibody acts like a flag, calling in immune cells to destroy the tumor. A drug called dinutuximab, which targets the GD2 ganglioside, is already approved to treat high-risk neuroblastoma, proving this concept works. We can take this a step further with antibody-drug conjugates, or ADCs. These link a TACA-targeting antibody to a potent chemotherapy drug, creating a guided missile that delivers its toxic payload directly to the cancer cell, minimizing collateral damage to healthy tissue. Second, we have TACA-based cancer vaccines. The goal here is to train the patient's own immune system to recognize these sugars as foreign and attack them. This has been challenging because carbohydrate antigens don't typically provoke a strong immune response. But researchers are making progress by linking the sugars to larger carrier proteins to make them more visible to the immune system, and by combining these vaccines with checkpoint inhibitors to unleash a more powerful anti-tumor response. Finally, there's adoptive cell therapy, most famously CAR T-cell therapy. This involves taking a patient's own T-cells, engineering them in a lab to recognize a specific target, and infusing them back into the patient. Scientists are now designing CAR T-cells that are engineered to recognize and hunt down cancer cells based on their TACA signatures. CAR T-cells targeting GD2, TNMUC1, and other TACAs are already in clinical development. An even more futuristic approach involves engineering immune cells to perform glycocalyx editing. Imagine arming an NK cell with enzymes that act like molecular lawnmowers, shaving off the mucins and sialic acids that form the cancer's protective shield. This strategy can breach the armor, allowing the immune system to finally see and kill the cancer cell beneath. The study of how cells use sugar has opened a thrilling new frontier in the war on cancer. We now see that the sweet coat on a cancer cell isn't just a disguise. It's a sophisticated weapon system. But this sweet deception is also cancer's Achilles heel. By continuing to unravel the complex language of sugars, we are developing a new generation of therapies designed to strip away cancer's armor, expose its identity, and let our own immune system deliver the final blow. The future of oncology is looking sweeter than ever. Thanks for listening to G Fun Facts Online.